Daily quality control of the MRM-MS platform is achieved with Kit 1A. This kit contains a plasma digest that has been spiked with an isotope-labeled peptide mixture, an isotope-labeled peptide mixture in buffer, and dilute formic acid. After rehydrating the two peptide mixtures with a specified volume of the formic acid solution, transfer their contents to autosampler vials and place them in an autosampler rack for automated LC-MRM analysis. Inject the labeled mixture in buffer for retention time scheduling and the plasma standard for performance monitoring. During the analysis, ensure accurate peak selection and integration, and then track several parameters, such as response and retention time over the course of a week. Kit 1B assesses the LC-MS system through the quantitation of 7 plasma proteomic standards. These must first be rehydrated with the supplied formic acid solution and then transferred to conical autosampler vials before LC-MRM analysis. From the standard curves, various assay performance attributes can be determined, which can then be compared to their reference values for accuracy assessment. Kit 2 assesses the performance of the entire analytical workflow. To begin, thaw the supplied plasma on ice while ammonium bicarbonate and other chemicals are being weighed. Once thawed, dilute the plasma tenfold with ammonium bicarbonate and place on ice until use. To prepare for the triptych digestion, place the ammonium bicarbonate buffer in 3 separate tubes, then add the diluted blood plasma, 10% sodium deoxycholate for protein denaturation, and 0.5 molar TSEP for disulfide bond reduction. After a brief vortex and centrifugation, incubate the samples in a 60-degree oven for 30 minutes. After this incubation, add iodoacetamide to each tube to alkylate the reduced sulfhydryl groups. Briefly vortex and centrifuge the solutions before allowing the reaction to proceed for 30 minutes at 37 degrees. DTT is then added to each tube to quench the unreacted iodoacetamide. Again, after a brief vortex and centrifugation, the samples are incubated at 37 degrees for 30 minutes to allow for completion of the quenching reaction. Rehydrate the supplied trypsin in ammonium bicarbonate, and then add a 10:1 aliquot to each tube. After a brief vortex and centrifugation, incubate the samples overnight at 37 degrees. Before the digestion is complete, rehydrate the supplied cis mixture and prepare a 10,000-fold concentration series. Vortex and centrifuge each mixture before preparing the lower concentration standards. After digestion is complete, pool the digests into a single falcon tube, then, in 7 separate Eppendorf tubes, place aliquots of the digest and aliquots of the different isotope-labeled mixtures. After a brief vortex and centrifugation, acidify the post-digestion mixtures with 1% formic acid and spin down the acid-insoluble surfactant for 10 minutes at ambient temperature. If the preparation is done correctly, the precipitate should be clearly visible. Desalt and concentrate the surfactant by solid phase extraction using syringe barrel cartridges and a vacuum manifold. After eluting the peptide mixtures into 7 autosampler vials, cover each tube with parafilm, freeze in a sample tray, puncture holes with a needle, then freeze dry the standards by overnight lyophilization. Upon sample rehydration with an aliquot of dilute formic acid, place the 7 standards in the autosampler rack for LC-MRM analysis. From the resulting standard curves, a series of performance metrics can be determined, which can be compared to the reference values in the kit to determine the accuracy of the entire analytical workflow.